Hello, everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss the battle between comb jellies and sponges for sisterhood to the rest of the animal kingdom. So, let's jump right in. At this point in our series, we have covered the bilaterians, animals that are bilaterally symmetric, at least at some stage during their life cycle, as is the case for echinoderms. The first 48 episodes were focused on members of this enormous group. Secondly, we covered the radially symmetric cnidarians in the previous two episodes. Bilateria, cnidaria, and possibly Placozoa are united under the clade Planulozoa. As Wahlberg et al. 2004 says, quote, possible synapomorphies of Planulozoa are the presence of endodermal myoepithelial musculature, septate junctions joining the epithelial cells, symmetrically arranged spermatozoan heads with a midpiece and similarities between the cnidarian planula larva and an acial worm, both of which are vermiform and have apolar development of the nervous system. Additionally, the cnidarians carry a set of several clustered Hox genes which are similar in arrangement and expression to those in bilaterians. Close quote. The uncertainty lies in the precise position of Placozoa. They were originally believed to be the earliest diverging animals, more distantly related to us than even the sponges. This was based on the fact that Placozoans are very simple animals. They look like amoebas at a glance, except for being multicellular. However, most genomic studies conclude that Placozoans, bilaterians, and cnidarians are more closely related to each other than any one of them is to sponges, but the exact phylogeny is disputed. Some phylogenies have placed Placozoa as sister to cnidaria and bilateria, and this total clade has been called Parahoxozoa because all these animals possess both Hox and Parahox gene clusters. If Placozoa is sister to cnidaria, then Parahoxozoa is synonymous with Planulozoa. If this relationship is true, then it would suggest that Placozoans descended from ancestors that were originally more complex, possessing tissues, muscle, and nerve cells. Just another reminder that evolution is not inherently directed towards increasing complexity. The jellyfish's tail dropped us off 590 million years ago in the Ediacaran period. We met the bilaterians, stem bilaterians, and cnidarians, but there are still a few groups left to encounter before we reach the sponges: Trilobizoa, Rangiomorpha, Arboriomorpha, and Ernietomorpha. They are all considered probable stem metazoans, meaning these are more likely to be closer to the aforementioned Parahoxozoans than they are to sponges, but a more exact placement for each relative to the others is unknown. Eumetazoa, sometimes called Epitheliozoa, is the clade that includes all animals except for sponges, which have differentiated tissues. However, whether this relationship holds is going to depend on issues we'll touch on later, so stay tuned. Trilobizoa is a clade of tri-radially symmetric benthic animals that have been interpreted as cnidarians or echinoderms, although it's now considered unlikely to be either. They lived from 565 to 546 million years ago, and the most well-known member is Tribrachidium. The name Trilobizoa refers to their dome-shaped bodies composed of three lobes called antemeres. The antemere shape varies across species. In Ancestor, each antemere is symmetrical, but in Albemarle, each antemere is elongated to one side. In Tribrachidium, this elongation is much more extreme. Viewed from above, there are three grooves, each leading to digestive cavities, and along the surface of the dome are uniform ridges that most likely held cilia, which move food to the grooves. Alternatively, in one specimen, there appears to be a Y-shaped structure interpreted as a mouth, but this feature is absent in all other specimens, so it could be a one-off artifact. A 2021 paper described a slightly curved trail nearly as large as the Trilobizoan that leads away from it, indicating that these animals could move, albeit very slowly. Based on these characteristics, the paper concludes that Trilobizoans are in the same paraphyletic grade of animals that includes ctenophores and cnidarians, but are not members of either phylum. Next, we turn to the frond-shaped animals, Rangiomorpha, Arboriomorpha, and Ernietomorpha. Collectively, they are called Petalonamae. They existed from 571 million years ago to at least right after the start of the Cambrian. Some may have survived until 516 million years ago if Stromativarus is a Petalonamid, or 508 million years ago if Thaumaptilon is a Petalonamid. Scientists have only their fossils to figure out their phylogenetic affinity, but none of those fossils can be well characterized. The first to be described back in 1929 was Rangia, a rangiomorph. It was also the first macroscopic Precambrian organism described. Rangiomorphs are unique among life extant or extinct. Their bodies display a fractal pattern. The frond is made of branching frond elements that are made of smaller frondlets, and the organism is anchored to the sediment by a holdfast. Arboriomorphs and Ernietomorphs are also frond-like with a holdfast, but they do not have the fractal pattern. All members of Petalonamae have been difficult to place, with their affinities ranging from sea pens to fungi to lichens to independently multicellular eukaryotes, but none of these are considered valid today. A 2018 paper by Jennifer Cuthill and John Henn argues that Petalonamae is sister to Eumetazoa, and a 2021 paper by Francis Dunn et al. Affirms this conclusion. They write, quote, "Our analyses reveal a highly connected and compartmentalized internal architecture exhibiting branch origination points that are topologically constrained. These data do not support models that suggest substantial plasticity in rangiomorph growth programs. We exploit this new understanding of the biology of Charnia to constrain the phylogenetic affinity of rangiomorphs to stem Eumetazoa, confirming a diverse Ediacaran history for this fundamental metazoan clade and demonstrating the capacity for members of the Ediacaran macrobiota to inform the timing and patterns of character acquisition in early animal evolution." Close quote. Because rangiomorphs were dwellers of the cold, deep sea, they must have been heterotrophic, but how they acquired food has been the subject of much debate. Typically, the rangiomorphs are seen as osmotrophs, meaning they take in dissolved organic carbon through osmosis. This is also what we said in our video on the Cambrian explosion 6 years ago. However, a 2020 paper by Nicholas Butterfield called this hypothesis into question. He proposed that these animals had water-filled compartments through which water flowed via ciliary pumping, allowing them to internally process food similar to sponges or cnidarians. Most Petalonamids, though, were wiped out in an extinction near the end of the Ediacaran, with only a few individuals straggling into the Cambrian. One more relevant piece of paleontology that has ignited debates has been the Duchantuo Formation, which spans 635 to 551 million years ago. This site has yielded a variety of microfossils, including embryos with a characteristic type of preservation called phosphatization, in which biological material is replaced with phosphate. Unfortunately, phosphatization has a size limit. Only up to 2 millimeters can be preserved, meaning these phosphatized embryos can only reveal so much about what their larger forms might have been. In 1998, Shuhai Xiao et al. Reported numerous organisms from the site. At least 8 taxa of cyanobacteria, 31 acritarch taxa, more on these momentarily, fragments of red and green algae, and potentially animal, even bilaterian, embryos. The conclusion that some of these microscopic fossils were animal embryos was immediately challenged, with some researchers suggesting the embryos were instead giant sulfur-oxidizing bacteria similar to Thiomargarita. Whether some of the alleged embryos are in fact bacteria could be the case. However, this assignment cannot be true for all of them. Part of the reason is that bacteria do not form large, preservable cysts with complex ornamentation. The Duchantuo microfossil Tianzuchenia, though, does have these features, meaning it is almost certainly a eukaryote. Acritarch is the generic label for a fossil eukaryote of uncertain affinities. So, since we don't know the exact phylogenetic placement of the Duchantuo microfossils, they are all acritarchs. Once we figure out what one is, it stops being an acritarch and becomes a member of its newly assigned clade. As mentioned earlier, the Duchantuo formation contains dozens of acritarch taxa, such as Tianzuchenia, Spiralicellula, Caviosphera, and Helicoforamina. Caviosphera is another multicellular acritarch contained within an envelope, and studies of its characteristics tend to conclude that it is at least a holozoan, the clade containing ichthyosporians, philisterians, choanoflagellates, and animals, if not an animal. Nailing it down more precisely is currently not possible. A similar holozoan affinity has been assigned to the spirally shaped Helicoforamina. Now onward to the period preceding the Ediacaran, the Cryogenian, which lasted from 720 to 635 million years ago. As one might expect, the Cryogenian period was a cold time. The start was marked by the Sturtian global glaciation event from 720 to 659 million years ago, and ending with another one called the Marinoan glaciation from 645 to 635 million years ago. Debates have raged over whether the Earth's surface was completely covered in ice during these periods, the snowball Earth scenario, or whether the equatorial regions remained unfrozen, the slushball Earth scenario. Either way, ice covered most of the continents. This was a radical shift from the preceding so-called boring billion, during which the climate remained rather stable. The glaciation events were caused in part by the movement of the supercontinent Rodinia. Everyone is familiar with Alfred Wegener's supercontinent Pangaea that lasted from 335 to 200 million years ago, but Rodinia appeared long before Pangaea, from about 1.23 billion years ago to 633 million years ago. One part of Rodinia, called the Franklin Large Igneous Province, or LIP, is typically invoked as a major reason for cooling. To understand this, we need to cover a process called the carbonate-silicate geochemical cycle. The first series of reactions involves chemical weathering of rocks. These reactions consume carbon dioxide, water, and calcium silicate to form a calcium ion, bicarbonate ion, and dissolved silicon dioxide. The products end up in the oceans, where a second series of reactions uses the calcium ions and bicarbonate to make calcium carbonate. This reaction is facilitated by various organisms, including the aragonite skeletons of the corals we mentioned in the previous episode. The formation of calcium carbonate releases carbon dioxide and water as waste products. Now, a key detail to keep in mind is that reactions of chemical weathering at the beginning used 2 carbon dioxide molecules per calcium atom, whereas the second reaction only returns 1 carbon dioxide molecule back to the atmosphere per calcium atom. The second carbon dioxide remains locked up as calcium carbonate. Thus, as the calcareous skeletons and shells of organisms end up down on the seafloor, this results in the decrease of atmospheric CO2 levels. If this continues unimpeded, there would be no CO2 left in the atmosphere. However, due to plate tectonics, the seafloor containing the calcium carbonate will eventually be subducted down into the mantle. Down there, the high temperatures and pressures allow the calcium carbonate to react with silicon dioxide, producing calcium silicate and carbon dioxide, which is released back into the atmosphere by volcanoes or hydrothermal vents, thereby completing the carbonate-silicate cycle. This geochemical cycle has often been referred to as Earth's thermostat, since it is subject to a self-stabilizing negative feedback loop. Chemical weathering is an important step in this process, but it is temperature dependent. At low temperatures, or when rocky surfaces are covered by ice, the rate of chemical weathering is reduced, and less CO2 is removed from the atmosphere. Meanwhile, volcanoes will still release CO2 into the atmosphere. The end result is a net increase in atmospheric CO2, and thus increasing the temperature of the Earth. But when the Earth gets warmer as a result, it increases the rate of chemical weathering, so more CO2 is removed from the atmosphere. Thus, over long periods of time, these two processes tend to balance each other out. Emphasis on long term. Unfortunately, the carbonate-silicate cycle is just too slow to counter the amount of CO2 we are dumping into the environment. Furthermore, temperature is not the only factor that affects the carbonate-silicate cycle. Rates of chemical weathering and volcanism can change by other means. Chemical weathering is enhanced when new mountain ranges are rapidly formed. A good example of this is the Himalayas, which formed during the Eocene when India collided with Asia. The chemical weathering of these mountains started a long-term cooling trend. A 2024 paper by Adriana Dutkiewicz et al. Points to both rapid chemical weathering and the fact that outgassing from mid-ocean ridges appears to have been significantly lower than usual during the Sturtian glaciation. So, the combination of silicate weathering and the very low outgassing of carbon dioxide caused the Earth's temperature to take a nosedive until glaciers clawed their way to the tropics. Eventually, Earth pulled itself out of the Sturtian glaciation after enough carbon dioxide accumulated in the atmosphere. In contrast, much less is known about the later Marinoan glaciation event, so the task is up to future geologists to figure it out. During the glacial periods, the ecology changed significantly from being cyanobacteria dominated to algae dominated. The difference between cyanobacteria and algae is that algae are eukaryotes, which are able to perform photosynthesis inside their organelles called chloroplasts. These chloroplasts used to be free living cyanobacteria but ended up inside eukaryotes via primary endosymbiosis. Eukaryotes with primary endosymbiotic chloroplasts are the archaeplastids, which include the Glaucophyta, Rhodophyta, and Viridiplantae, the latter of which would give rise later to the land plants. But shenanigans with chloroplasts doesn't end here. It turns out that other eukaryotes also got chloroplasts, but via secondary or even higher order endosymbioses, meaning a whole eukaryote possessing chloroplasts ended up becoming the chloroplast inside another eukaryote. Later, irradiation in algae with secondarily endosymbiotic chloroplasts would include diatoms, foraminifera, radiolarians, and coccolithophores in the Phanerozoic. If you want to know more details about the chloroplasts and how they are passed from eukaryote to eukaryote, please check out our video on passing chloroplasts from 4 years ago. In the geologic record, we observe a major shift from bacterial steranes to an overwhelming abundance of algal steranes, indicating the bacterial to eukaryotic phytoplankton transition occurred between the Cryogenian glaciation events. But the earliest evidence of archaeplastids, such as Raphitasmia, comes from 1.6 billion years ago. So what took algae so long to get established? One suggestion is that the Sturtian glaciation event cleared the ecological deck. Whereas before, life survived in warmer climates nearly globally, the glaciation served as an evolutionary bottleneck for most clades. Refuges for organisms during the glaciation events were, aside from the possibly open equatorial ocean, likely cryoconites, which are microbe mineral deposits on glaciers. Dirt and soot can absorb solar radiation, warming up and melting localized ice to provide fresh water. Microbes can also produce biofilms and cryoconites to increase their habitability. On modern glaciers, cryoconites host proteobacteria, bacteroidetes, cyanobacteria, protozoans like choanoflagellates, whom we'll meet next time, microalgae, fungi, and animals like rotifers and tardigrades. The 2022 thesis of Jasmine Miller found, quote, Antarctic cryoconite was resilient to 2 snowball Earth low-latitude conditions, daily day-night cycles, and daily freeze-thaw cycles. Under both conditions, the cryoconite communities were able to reach a growth phase comparable to that of control conditions of constant light and temperature, close quote. When the glaciers began to melt at the end of the Sturtian, massive amounts of fresh water would have been dumped into the oceans, causing hyposaline conditions and favoring the expansion of formerly freshwater lineages of cyanobacteria and algae. Indeed, the dominant marine cyanobacteria in today's oceans, Senecacoccus and Prochlorococcus, originated in terrestrial freshwater environments before colonizing marine environments about 700 million years ago, and the closest living relative of the chloroplasts is Gliomargarita lithophora, which is found in freshwater environments. Furthermore, glaucophytes, which are sister to red and green algae, are found in freshwater environments, and the green prasinophytes are descended from freshwater ancestors. Another hypothesis for the domination of algae involves low levels of phosphate. Cyanobacteria dominate under poor nutrient conditions, so low levels of phosphate allowed them to prevail over the algae. Low levels of phosphate seem to have been maintained by anoxic deep water conditions, which removed phosphate from surface waters through co-precipitation with iron minerals. Without enough phosphate, primary productivity was decreased, as was carbon burial, eventually decreasing the amount of oxygen in the atmosphere. Less oxygen in the atmosphere meant less oxygenated deep sea regions and so on, feeding the cycle. When the Sturtian glaciation occurred, nutrient-rich ground rock debris was transported to the oceans, massively increasing the amount of phosphate in the oceans. As carbon dioxide levels and therefore temperatures increased during deglaciation, the weathering of glacial sediments would have added even more nutrients to the oceans, skyrocketing primary productivity. Parallel with the rise in algal steranes during the Cryogenian greenhouse interlude was the rise in Rhizarian steranes. Rhizaria today includes numerous species of amoeboid protozoans, foraminifera, and radiolarians. In other words, eukaryovorous predators blossomed along with algae. This potentially fueled an evolutionary arms race that led to the emergence of animals. Indeed, the earliest current evidence of animals dates to about 650 million years ago in rocks from Oman. That evidence of animals comes from steranes, specifically ratios of 24 isopropyl cholestane to 24 N-propyl cholestane. While these steranes can be produced by sponges, they can also be produced by two species of unicellular marine pelagophyte algae. However, the ratios of these molecules produced by the algae and sponges differ substantially. As one paper explains in its supplementary information, quote, for pelagophyte algae, the 24 isopropyl cholesterol is a minor byproduct of the biosynthesis of the much more abundant sterol compounds containing a 24 N-propyl cholestane skeleton. It is an aberration of an enzymatic process intended to produce 24 N-propylidene cholesterol and related structures, and does not constitute a biosynthetic capacity to make abundant 24 isopropyl cholesterol given optimal growth conditions. Close quote. In 2014, the paper giving the early fossil record of sponges a squeeze argued that since the sterane is known to be produced by other organisms, its mere existence isn't enough to validate the sponge hypothesis. Further, this paper repeated the claim that a group of symbiotic bacteria called poribacteria could also make the molecule. Let's take the latter argument first. This is simply wrong. The notion that poribacteria could make the sterane was evidently due to a genome assembly error. As for the former, phylogenetic analyses of the distribution of sterane production among eukaryotes indicate that pelagophytes need 3 copies of a gene called carbon 2428 sterol methyltransferase to make 24 isopropyl cholestane, and sponges have 2. Using molecular clock estimates, a team of researchers showed that the duplication event for the sponge gene overlaps in time with the steranes, while the duplication of the pelagophyte gene occurred substantially later in the Phanerozoic, at least 100 million years later. Finally, on this point, in 2018, it was revealed that the sterane 26-methylstigmastane was also found in the Oman rocks, which is a compound unique to demosponges. Though some protists may make precursors to both 24 isopropyl cholestane and 26-methylstigmastane, none are known to make both compounds, and experiments attempting to chalk up the presence of the molecules to inorganic processes didn't realistically approximate natural conditions at all. Thus, researchers have tended to regard the presence of those steranes starting at about 650 million years ago as the earliest evidence of animals. To give an overview of animal evolution in the late Precambrian, the first evidence of animals dates to 650 million years ago. Stem metazoans, like the rangiomorphs, show up 571 million years ago. Stem bilaterians, like the phylum Proarticulata, whom we met in the jellyfish's tail, are evident 567 million years ago. And crown bilaterians, like Icaria, appeared at least 555 million years ago. This seems like every modern animal did not just suddenly appear out of nowhere in the fossil record. Now, after that long introduction, we can meet the 2 phyla on which this tale focuses: Ctenophora, the comb jellies, and Porifera, the sponges. We'll take the ctenophores first. Comb jellies are named for their 8 rows of cilia which they use to swim. They do not use rhythmic contractions of a bell like jellyfish. Another difference between them and true jellies is that comb jellies swim mouth first, but like jellyfish, ctenophores capture zooplankton for food. Unlike cnidarians, ctenophores do not capture their food with cnidocytes, but stick to it using cells called colloblasts. The colloblasts are often arranged along tentacles that drift behind the swimming animal. Also, unlike cnidarians, ctenophores have anal pores that allow small bits of food to be excreted, although most undigested food is excreted through the mouth. Ctenophores and cnidarians both have a diffuse nerve net and a jelly-like mesoglia. Most ctenophores are globose or ovoid, although Venus's girdle, Cestum veneris, is ribbon-shaped. And bizarrely, ctenophores formerly assigned to the now polyphyletic order Platytenida are flat, crawling along the seafloor like slugs. Cnidarians do not have muscle tissue, but they do have myoepithelial cells, while the ctenophores have both an epithelial net of muscles and mesoglial myocytes. Additionally, the neurotransmitters used by cnidarians and bilaterians, such as acetylcholine, glutamate, and GABA, are different from those used by ctenophores, which are mainly peptidergic synapses. And finally, neuronal specific genes are very different in both phyla, with ctenophores lacking major genes expressed in cnidarians. This indicates that the nervous systems are analogous, having evolved independently in the phyla from a common eumetazoan ancestor which lacked a nervous system. Ctenophores have an apical or aboral organ that is structurally different from the apical organ of cnidarians and bilaterians, meaning that the organs are non-homologous. Another reason for the lack of relatedness in this organ is that the cnidarian apical organ is involved in larval metamorphosis, whereas ctenophores skip the larval stage entirely, going straight to the juvenile. There is also a dome of cilia covering statocysts on the ctenophore that are lacking in cnidarians. A few fossil ctenophores are known, and they bear characteristics unknown in any modern species. For example, stem ctenophores like Gemectina had an internal skeleton and a prominent circumoral structure called the oral skirt. From there, the stem ctenophores reduced their skeleton until it was just a capsule over the aboral organ that can be seen in the Cambrian stem ctenophore, Tenor rhabdotus. The aboral organ capsule was then lost, which can be seen in Thalassostaphylos, and following this taxon, the oral skirt was lost and the number of ciliary bands was reduced. Pulling out a bit further, the stalked sessile Scyphusoctum from the Cambrian Burgess Shale is sister to the clade containing crown and skeletonized ctenophores. This odd animal has large ciliary structures that belie its true affinities. At the base of total group Ctenophora is the family Dinomycidae, which contains such members as the namesake Dinomycius and Xiangguangia. Like Scyphusoctum, they were all stalked and sessile, but they also had tentacles that ringed the mouth. When comparing the stem ctenophores against other animals, Yang Xiao et al. Found that they were sister to the polyp-bearing cnidarians, with poriferans being sister to all animals. Now let's move to the other relevant phylum, Porifera, the sponges. Sponges are the only animals that lack true tissues and organs, being composed instead of two cell layers sandwiching a jelly-like material called mesohyle. Even though these cells may appear tissue-like, they are not bound tightly by cell-to-cell connections nor a basal lamina. Sponge pores, called ostia, in the sponge allows water to flow into the animal. And ciliated cells called choanocytes, which indeed look superficially similar to choanoflagellates, pick nutrients out of the water. We'll talk more about these cells in the next video. Nutrients are digested intracellularly because sponges lack a digestive tract. They also lack a nervous system. However, sponges are capable of contracting the cells that make up their outer surface, called pinacocytes, to relieve pore blockages caused by sediment. Some have humorously termed this motion sneezing. Once water is processed by the choanocytes, it flows out through a big pore called the osculum. Internally, sponges can be arranged in a few ways. One structure is called asconoid, where there is a single layer of choanocytes through which water flows. Another is coenoid, where the sponge has pockets lined with choanocytes to increase the amount of water that can be processed. And finally, there are leuconoid sponges that have multiple connected chambers for processing water. Another type of sponge cell is the archaeocyte or amoebocyte that moves around in an amoeba-like manner and are totipotent, meaning they can transform into any cell type. Sclerocytes secrete mineralized spicules that form the skeleton of sponges and protect them against predators, and just like all other animals, sponges have the ability to produce both sperm and eggs. There are 4 classes of sponges: Calcarea, Demospongiae, Hexactinellida, and Homoscleromorpha. First, calcareous sponges are named for their calcium carbonate spicules, and this class contains some 800 species. Second, Demospongiae, by contrast, contains some 8,800 species, making it by far the largest class of sponges. Demosponges have spicules made of the protein spongin, silicon dioxide, or both. Third, Hexactinellida are known as the glass sponges because their silica spicules form a lattice network. This class has about 500 species. Fourth, Homoscleromorpha consists of 136 species and has one particular feature that sets it apart from all other sponges: a basement membrane. On the basis of this feature, and in conjunction with some limited genetic studies, some zoologists argued in the early 2000s that homoscleromorphs were actually more closely related to other animals than to other sponges, making porifera paraphyletic. This suggestion has evaporated with larger datasets, finding that homoscleromorphs are sister to calcareous sponges. There are numerous fossils of sponges in the Phanerozoic, but sponge fossils in the Precambrian have been much more contentious. As discussed earlier, sponge steranes from the Cryogenian currently represent the earliest evidence of animals, and Eocyathospongea, hailing from the Duchanteau Formation 602 million years ago, is considered to be a stem sponge. Since sponges are largely soft-bodied and their spicules can be destroyed easily, hunting for Precambrian sponges, or ctenophores for that matter, is a difficult task. Alright, the stage is set and the players are in place. The question on everyone's mind is, who is sister to the rest of the animal kingdom? The hypothesis that sponges are sister to all other animals is called porifera first, while the hypothesis that ctenophores are sister to all other animals is called ctenophora first. Tracing a history all the way back to Ernst Haeckel in the 1860s, porifera has been traditionally considered sister to all other animals on the basis of its multicellularity and phagocytic capabilities, but lack of true tissues. It just barely squeaks in as an animal. In fact, some phylogenies considered sponges a phylum separate from animals altogether, being independently multicellular protists, as can be seen in Preston Cloud's 1978 book Cosmos, Earth, and Man. This hypothesis has never been consensus, though. As for ctenophores, their phylogenetics has an equally fraught history. Originally classed by naturalist Georges Cuvier in the created group Radiata, along with Cnidaria and Echinodermata, all these animals share radial symmetry as adults. But as we saw back in the ragworm's tail, echinoderm larvae give away their true affinities. They are bilaterians. However, broad similarities between ctenophora and Cnidaria often kept them together as the clade Coelenterata in the 20th century. Then DNA entered the picture. That 2004 paper by Andreas Wahlberg et al. That we cited at the beginning investigated the position of ctenophores with respect to bilaterians and cnidarians. They used 528 animal and outgroup small subunit ribosomal RNA sequences, including almost 120 sponge sequences, finding that sponges are the sister group to all other animals, and ctenophores are sister to Cnidaria plus Bilateria. However, a 2009 paper by Hervé Philippe et al. Revived Coelenterata with a dataset of 128 different protein-coding genes. The same year, Andreas Hejnal et al. Interrogated the placement of the acoelomate flatworms we also met in the ragworm's tail using expressed sequence tags, which are segments of the complementary DNA of genes that are transcribed from 94 animal and outgroup taxa. This study found not only that ctenophores are sister to all other animals, but also that sponges are polyphyletic, not just paraphyletic. A 2014 paper by Casey Dunn et al. Cited a series of genetic papers in support of ctenophora first. In 2019, Christopher Laumer et al. Used 422 genes in 195 taxa to test broad relationships among Animalia. The paper writes, quote, "Remarkably, using this matrix, we find no particular support in any of these conditions for either porifera or ctenophora as the sister group to the remaining metazoa, unlike the strong support claimed in many recent analyses addressing this particular issue. While the combination of both Dayhoff-6 recoding and distant outgroup pruning does increase support for porifera in this position, the posterior probability for this split is still well below most reasonable significance thresholds. Given that other matrices we have analyzed under these conditions have recovered strong support for either ctenophora or porifera as the deepest splitting animal phylum, it would appear that the effects of removing distant outgroups and using reduced amino acid alphabets on this problem are matrix specific, contradicting the assertion that these approaches, in general, lessen systematic error and reveal the true phylogenetic signal in the data. Close quote. A 2021 paper by Redmond and McLissett found support for porifera first and noted, quote, while phylogenomics has revolutionized our understanding of the tree of life, commonly applied phylogenetic models do not account for a wide array of factors that are known to cause positively misleading phylogenetic estimates when overlooked. Such factors involve the non-independence of sites, variation of the substitution process across sites and taxa, as well as site-specific variation over time of the substitution rate and/or process. Close quote. Other papers have agreed that using different phylogenetic methods reveals different topologies. A 2017 Nicole King and Antonis Rokas paper points out that site homogeneous models give support for Ctenophora first, while site heterogeneous models give support for porifera first. For reference, site homogeneous models assume that any amino acid can be equally substituted for any other amino acid, which we know is not true in a number of cases, while site heterogeneous models do not make this assumption. King and Rokas state, quote, consequently, site heterogeneous models are better descriptors of biological sequence evolution than site homogeneous models, but are computationally much more costly, close quote. Other potential problems include long branch attraction within Ctenophora, an issue that we discussed in the Gibbon's tale. The common ancestor of ctenophoras appears to have lived in the early Carboniferous, some 350 million years ago, while their common ancestor with other animals lived at least 650 million years ago. This suggestion was made in a 2020 paper by Pashalia Capley and Maximilian J. Telford, saying, quote, long branch attraction can be exacerbated by the use of substitution models that do not account for heterogeneities in sequence evolution, such as non-homogeneous rates of substitution between alignment sites or heterogeneities in the frequencies of amino acids across the alignment, close quote. The team found that long branch attraction and site homogeneous models are two major reasons for Ctenophora first. Another set of issues includes strategies for identifying orthologs, the level of tolerance for missing orthologs, the choice of a non-animal route for the phylogeny, and rules for excluding fast-evolving sequences. These issues, though, are much less straightforward. In 2019, Klaus Nielsen authored a paper in which he argued for porifera first purely on morphological grounds. He points out that endoderm, ectoderm, and extracellular digestion are shared among all metazoans. Therefore, if the Ctenophora first hypothesis is true, that requires sponges to lose both germ tissues and revert to intracellular digestion, which seems exceedingly unlikely. Alternatively, Ctenophora first could imply that the digestive system evolved twice, once in Ctenophora and once in Planulozoa, which also seems unlikely since both share the same genes for this system. And that's the sponge's tale. Systematic phylogenetic errors and differences in methodology seem to be the explanation for Ctenophora first, while morphology and more realistic phylogenetic methods tend to support porifera first. In the next episode, we're going to look at the series of evolutionary changes that resulted in ancestral animals. So, thanks for watching, and we'll see you all next time.